So today we are going to learn about PCR. So PCR stands for polymerase chain reaction. Well, why do we use PCR? So basically PCR is used for DNA amplification. What it means is that it is used for replicating new strands, or in short, generating a lot of strands. So I mean, why is it required, right? Let me give you an example. In a crime scene we have very limited amount of DNA present, right? So if we want to use it in the forensic laboratory for research purposes, I mean that small amount of DNA will be finished off, right? And we need a lot more amount of DNA. So with the help of PCR we can generate a lot of new copies from that less amount of DNA. So this is one of the applications. So in order to amplify the DNA we need a lot of requirements, right? So the requirements also is known as the master mix. Right. So what are things that comes under it, first of all, is the DNA polymerase. Okay, over here particularly we will use Taq polymerase, which is extracted from a bacterium called Thermus aquaticus, which thrives in extreme conditions, okay, high temperature conditions. Then comes DNA templates, right? Without this there is no use of doing PCR. Third is the DNTPs, okay, deoxyribose nucleotriphosphate. Then comes various divalent cations, for example Mg2+. Then the buffer solution, etc. Okay, so these all requirements are needed in order to perform the PCR. Now let us understand the whole mechanism. So now let us understand the whole mechanism, the whole process, all 3 steps which are involved in the PCR. So the first step is denaturation. The second step is primer annealing. The third step is primer extension. Well, we will look into details on it. We won't go in a lot of detail. I'll explain a brief overview of it. So in denaturation what actually happens is we have the double stranded DNA, right? Now in vivo various various different proteins and various enzymes are already present in it, which actually separates both DNA strand. For example, single stranded binding proteins, the helicases, the topoisomerases, all enzymes are involved. But in vitro we don't have that all, right? We have limited amount of things. So here we are going to use temperature in order to denature the DNA strand, right? We have this DNA strand. Now in order to break the hydrogen bonds, we need to apply temperature of about 92 degrees to 95 degrees, right? So at this temperature, all the hydrogen bonds will break from the bases and we'll get two separate single strands of DNA. So what happens in denaturation? We break the hydrogen bond and we get two separate DNA. The time taken over here is about 30 seconds to 1 minute, and it varies on the DNA strand which we have taken. So the next step is primer annealing. So I have highlighted a name primer annealing. In general, the step is called annealing, but here I have even mentioned primer, because without the primer we won't be able to synthesize the new strand along with it, right? So here we will be taking two type of primers. One is the reverse primer, second is the, guess what, forward primer. Right. So we have this two strand, one is 3' to 5', another is 5' to 3'. So the RNA primer will read at the 3rd prime, okay, synthesize 5' to 3'. Similarly, the reverse will read over here, synthesize from 5' to 3'. Right. So now, according to, like, from the DNA which we have taken from the sample, now the primer will also vary, right? So the temperature which will take over here also varies from the primers, right? And we measure it with the help of a rule called Wallace rule, right, which is like A plus T into 2 degrees Celsius plus G plus C into 4 degrees Celsius. This will give us the temperature of that particular primer, right? So we will be able to easily calculate the temperature of that particular primer from this method, and will take about 1 to 5 degrees Celsius less than the Tm which we have taken from that particular primer, right? So this actually happens in the primer annealing. So here Tm stands for melting temperature. At this temperature, the particular primers will no longer be able to function. Okay, so that's why we will take about 1 to 5 degrees less than the Tm temperature. So moving to the next step is the extension step, right? So like in general, like before moving to that, in general, if we talk about the primer temperature, it's about like it can vary from 55 degrees Celsius to 70 degrees, or it can even vary even less than this, right, according to the primer which we have taken. But let us assume we have taken a 55 degrees Celsius over here, right, and the amount of time required to the whole process to take place is about 30 seconds to 1 minute, again same. So now let us move to primer extension. So what happens in the primer extension is that the DNA polymerase acts and joins the various nucleotides, DNTPs, extend from the primers, right? So let us understand from the diagram. We have these two strands. We have these two reverse and forward primers. So now the DNA polymerase or as we have discussed that it is the Taq polymerase which is extracted from Thermus aquaticus. Right, it will keep on joining these nucleotides in both the direction, right, and this temperature range from 72 degrees Celsius to about 80 degrees Celsius, right. The time taken for the primer extension is about 1 or 2 minutes. So we have covered denaturation, primer annealing and the primer extension. So this three process completes one cycle, right, from denaturation to here and again from here to here, right. So about in order to perform a complete PCR, but it generally varies from 20 to 40 cycles, right. Like the amount of DNA which we require, if we have less amount of DNA, we'll take up to 40 cycles, but if we have already enough DNA and we want less amount of DNA to be amplified, we'll take about 20 cycles. So, and even apart from this, there are various different type of PCRs used, which we'll talk in different another time. Okay, so as we have discussed all the three processes, right, denaturation, the primer annealing, the primer extension. Now where does it take place, right, we'll have to even think about that. So there is a machine called thermal clear, right. So it's a quite expensive machine where you perform the whole PCR, okay, and actually there are special type of tubes involved, okay, in order to conduct this. They are known as PCR tubes. They are generally small in size than the normal ones, right. So that's it. Thank you.